Awesome. Thank you, everyone. Thanks for joining me bright and early on a Friday morning. My name is Esha. I'm a bioinformatics engineer at Seqera. I work on the scientific development team. We're a team of diverse bioinformaticians working with users like you, trying to understand your challenges, your workflows, and your Friday afternoon frustrations. Today, I want to talk a bit about scale. Not just your kind of typical and obvious kind where bioinformatics needs more compute, you need to scale with larger data, but real scaling happens on multiple axes: your code quality, your infrastructure flexibility, and your entire project lifecycle. So modern bioinformatics doesn't just scale up, it scales in every direction, and that's kind of what I want to talk about today. So last year, my wonderful colleague Rob Syme, introduced us to the journey from small Nextflow to big Nextflow, your code evolution from a very tightly knit main.nf script towards a portable and well-tested production pipeline. And what we've essentially learned and continued learning since then is that the Nextflow journey is just one of the axes. It doesn't just stop and end at your pipeline. Essentially, when we look at expansion from small Nextflow to big Nextflow, you essentially accumulate a lot of complexity across multiple areas. The first being your teams. It's no longer just you developing a pipeline or just you looking at your code. It might be other bioinformaticians, wet lab scientists, computational biologists. Your software, it'll maybe start with a couple tools and you'll scale into many, many more, all with their own dependencies and requirements. And as you go further along the scale, compute. You now need more flexible compute from going from your local laptop to maybe an HPC, maybe also the cloud. And your data. This isn't unfamiliar to us, but you start with maybe some test data, and now you need to be running your workflows on real-world data sets. And this continues on. We'll also encounter challenges with visualizations. It's not just enough to generate some results and store them in an S3 bucket or on a file system. Those results need to be explorable. And then lastly, containers. As you start to containerize your tools, you're maybe updating the tool versions and generating a bunch of different images that you're using within your workflows. So these complexities eventually just add up, but they're all essentially interconnected. And what I essentially want to do is organize these challenges into 4 connected axes. And they all scale in different directions. The first one being your code. And when we think about kind of your daily challenges, your code means essentially your development journey from that small Nextflow pipeline into something more kind of production ready, but with faster iteration. Your teams, you're not just writing more code, you're coordinating with more people. Each of those folks on your team need different interfaces that work for them. And your compute, your infrastructure needs to scale. Infrastructure also needs to adapt from going from a pipeline that's on your local MacBook to being able to run routine analysis maybe on an HPC. But what happens when you're limited on your cluster? You need to be able to scale into the cloud. How do you do that quickly and effectively? And then lastly, your projects. You're typically juggling multiple projects, and for each, you're maybe running multiple pipelines, different versions, multiple analyses, tracking exactly how you're doing said analyses. So reproducibility is very key. And essentially, you'll notice that each of these connect. Clean code enables reliable projects and analyses. Projects demand flexible compute based on the data that you get. And overall, you also need to be able to coordinate with your teams. So each axis is essentially affected by the others as well. What I want to do is show you how the Seqera ecosystem addresses challenges when you scale across all these axes, not as isolated features, but as an integrated platform. So before we dive into the solutions, let's be real about the challenges that we face at each of these axes. The first one being your code. What started out as a pipeline that you just wanted to get working, unfortunately, doesn't work anymore. Those shortcuts come back to haunt you. Your teams, what was once a beautiful pipeline may become a black box to everyone else who's also trying to collaborate on that analysis. And unfortunately, that leads to communication breakdowns between maybe the dry lab and the wet lab. Your compute challenges, we've all seen this before. Infrastructure eventually becomes a bottleneck. And lastly, your projects. The classic question of, how did you run this? And then followed by, it worked on my machine just fine. These can become recurrent and eventually leads to breakdown and prevents you from actually getting green check marks and getting to the results that you need. The key insight is that these aren't unsolvable problems or challenges, and that's exactly what we've been building for and what we've been talking about yesterday as well. And essentially what I want to show you is that we've seen the challenges, the roadblocks at every single axis, and these compounding problems over time will slow down your science. And the Seqera ecosystem has been built specifically to address each one. So let's see how we can kind of tackle these challenges. For our code specifically, we now have AI-assisted development with Seqera AI and the GitHub Copilot integration, powered by the language server, and as well as being able to do so in studios. For your team, one platform for everyone to collaborate on analyses, data, and compute, also providing custom visualizations and an easier method for everyone to access large data in the cloud. For your compute is now made easier with Seqera Compute. Getting access to running on AWS Batch within a couple of clicks makes it much easier to scale than ever. And lastly, your projects, reproducible analyses for each project all in one place on Seqera platform with all the tools you need to automate, debug quicker, and develop, getting you to the green check marks that you need. So what I want to do today is kind of frame the challenges that you'll see at each axis and how you can kind of overcome them using the Seqera ecosystem. And what I'd like to use is a scenario that we might all be familiar with, but just for the sake of the demonstration, let's say that you're working on some single-cell RNA-seq data, and your colleague Bob, who looks oddly very familiar to Ted Lasso, drops this Slack message to you at 3 p.m. on a Friday. There is some single-cell kind of aging pancreas data available for you to start running and analyzing. It's just a pilot data set. Unfortunately, when your colleague tries to run it, they encounter errors in the first process of the pipeline with command not found errors. So nothing necessarily new. To add to it, you get an additional message very close to 5 p.m. from another colleague, Jim, basically saying that they're unable to visualize their data in cell by gene and they need your help. The cherry on top is that there's an even larger data set that is waiting for you to be analyzed as well, all ideally by Monday. So, okay, this is a really fascinating study. What we're going to take a look at is cells from donors aged 20 to 70, investigating how aging drives cellular disorder. You have a pipeline. It's going to be doing batch correction, clustering, cell type identification. The workflow's there. You have access to an HPC. Sounds pretty straightforward, right? Wrong. You're hitting workflow errors the minute you try to run your pipeline. And when you do try to run your pipeline, you're hitting these unfortunate out-of-memory errors as well. Should you try to scale your workflow and start submitting jobs to your HPC, you're hitting queue limits, and your jobs are basically stuck in pending and submitted forever. And if you're brave enough, you will try to transition into the cloud, but unfortunately, you'll start drowning in IAM permission errors. So this is almost like the perfect storm. Urgent science meets infrastructure chaos, and there's critical research, but you're blocked by so many technical, compute, data barriers. This probably sounds familiar. Don't rage quit yet. It's fine. We'll figure it out. We're going to turn this disaster into a success story live. All right. I'm going to transition over into my laptop now. All right, so what you should be seeing on my screen is essentially a VS Code IDE. And what I first want to talk about is how we can scale your code. So what I have here is a very kind of tiny workflow. Let me show you this way. Essentially, this rough prototype Nextflow workflow will process single-cell RNA-seq data, count matrices to generate outputs for visualization. It works locally, kind of, but it's messy, and that's fine. When you're developing, you're not necessarily optimizing for the cleanest kind of production code. So the first thing we want to tackle is our colleague let us know that when they even tried to start running the pipeline, they run into command not found errors for the first process, this unify process. Luckily, through the new kind of Seqera AI sidebar, I can very easily navigate through the various processes within my workflow. This is probably easier done when you have 6 processes, but imagine as I continue on to build this pipeline, navigation to 20, 30 processes can be quite difficult. So what I'll do is I'll open up this first process, and taking a look at it immediately, I think I can figure out why they're running into those errors. They're probably running this workflow with Singularity or Docker, and there's no kind of container definition for actually specifying how this process should be run and what tools. That's fine. I mean, I could sit there writing a Docker file, building a container, but that's probably going to slow down my work quite a bit. And luckily yesterday, Sasha introduced us to this wonderful kind of wave integration where instead of me doing all of that manually, I can actually use Seqera AI to build me a wave container with the tools that I need. So what I'll do here is next to the process, I'll click on this wave icon. And Seqera AI is going to take a look at the process, find the packages that I need, and essentially give me a container at the top here. Now, all I can do is essentially drop this in. And that has just saved me about 30, 40 minutes of time. Instead of building a container, I've kind of gotten one out of the box, built by Wave and provided to me via Seqera AI. So now this should allow me to run this process within a container with all the tools and dependencies I need. This one in particular runs and data and also requires PyYAML, so luckily I was able to get these kind of out of the box. So what I can do now is start running the workflow, and hopefully it works. What we've also added in, just kind of out of best practice, is some sort of minimal test profile so I can check that the pipeline actually works. So what I'll do is actually run that now locally. I ran into a syntax error. Full transparency, I baked this in, but the whole point is that I can now use Seqera AI to actually help me figure out what went wrong and maybe even propose a fix. So what I'll do in this little sidebar where I can ask Seqera AI a bunch of questions is I've probably run into some sort of error and silly mistake that I made in my config. That's fine. Let me ask Seqera AI exactly what went wrong. My workflow failed with this error. Can you propose a fix? All right, silly mistake on my part. I essentially provided the wrong syntax for the resources that I was specifying for a process with this particular label. That's fine. Luckily, Seqera has my back and can make changes in line for me. Awesome. So now when I try to run my workflow, I shouldn't run into any issues. But while I'm here, I might as well kind of clean up my code as well. And luckily, I don't have to do this kind of manually. We have a wonderful new nextflow lint command. So what I will essentially do is run nextflow lint format. I'll sort the declarations. And why not include the new Harshal alignment flag? So this is proof that if you're obsessive enough about code formatting, you might get a flag named after you. All right, so I'll lint. And I've cleaned up my code, which is quite nice. I should be able to start running this now, so let's go ahead and do that. I hit an error. OK, let's figure out why. All right, so unfortunately, my workflow failed because the requirement for running the first process was 10 CPUs, and my local machine only has 8. See, the problem is that as development scales, we hit the limits of our local environment. I want to test this pipeline on this test data set, but eventually the actual pilot data as well. And my MacBook simply just can't cut it. It doesn't have the memory or compute to handle this. And that's fine. I could now set up an EC2 instance in AWS, reconfigure a development environment, install Nextflow, install Docker, all that. But that's going to cut down the time that I have by quite a bit. Instead, what I can do is transition into the platform. What we have here is essentially a Studios environment that I've created earlier that runs VS Code. What it also does is mount in a bucket where that pilot data that I'm going to have to analyze eventually is also stored. And I can make that available to me in this VS Code development environment using Fusion. So what I'll do is continue on. I'll connect into this. And this looks basically the same as our local development environment. The only difference now is I have access to data that lives in the cloud that I continue to use for testing and analysis. And now I'm not limited by the amount of resources I have on my local MacBook. So we'll keep on going. I'll run my workflow now here. And it's not only just about kind of performance. Think about it in this case. When we were talking about teams and collaboration, I can also now, whoops, I can't use Docker, let's use Podman. For now, we'll use Podman. It's also nice to kind of demonstrate different container engines you can use. In the future, we'll be able to use Docker. And so what I was referring to earlier is that it's not just about kind of performance, but collaboration as well. And now I'm enabling other bioinformaticians, maybe even scientists, to collaborate in the same VS Code environment, take a look at kind of the evolution of my code, and break down some of those silos that we were referring to earlier. So this will keep running, and luckily now I'm not limited by the CPUs on my local laptop. What I could also do is, given that I have access to that pilot data that is stored in S3 in this environment, I could actually start running that within this studio as well. We've given it a profile called pilot, and I can kind of go ahead and just run that. The problem is, though, I'm running on a single VM, so I'm going to run each task consecutively, which is not ideal, especially if I start scaling out to that 50K data set. And that's fine, but it still kind of brings and demonstrates that barrier for scaling your compute. So this is probably not the best solution to kind of run a larger data set. Instead, what we can do is use some of the additional compute options that are available in the platform. What I'm going to do is go into compute environments. So to solve this compute challenge, we know that our HPC queue is probably not going to cut it, and that's fine because essentially Seqera offers kind of different compute environments depending on what your needs are. And I want to quickly talk about those. So if we go into adding a compute environment, I have a couple different options available to me. What you just saw right now was a Studios environment that was running on our new kind of cloud VM compute environment offering. What that means is, as a bioinformatician, if I want to do some quick prototyping, spin up a studio, do some development on small data set, and not have to go through setting up that development environment and sandbox, I can use this new AWS Cloud CE to do that. It'll allow me to spin up an EC2 instance, hop into the environment, and either run a pipeline or continue on with development. And it's also nice because my DevOps team will appreciate that in that it's very easy to govern, it's kind of contained, and they can define exactly where those EC2s will run. If I wanted more kind of scale, as in I need to run tasks in parallel, a single EC2 isn't going to cut it, and that's where kind of Seqera Compute comes in. What I essentially have access to now is a managed solution for running jobs on AWS, without having to worry about any of the infrastructure setup and IAM rabbit holes. I essentially can run data at scale with urgent analysis and not have to worry about understanding what goes into setting up that infrastructure. We'll do this one right now. I'll set this up and we'll use this for running our pipeline. So I'll say this is single cell analysis. I'll select Seqera Compute, and all I have to do is also define a region, probably where my data is. You'll see that within 2 clicks, I'm able to now start running a pipeline on AWS Batch. I didn't have to worry about IAM roles, launch templates, setting up queues, anything of the sort. And essentially what that allows me to do is basically scale without any limitations, but I'll also know that there's a budget in place and I won't go over it in any problem. The last option that is just worth mentioning is AWS Batch Forge. So this hasn't gone away. This is kind of the option for full configurability when you want to tie in your own compute and essentially define exactly what that will look like, what kind of instance types you want, maybe networking requirements that your DevOps team has put in place. So now that we have different options for scaling our compute, let's go ahead and launch that workflow with the pilot data using the new compute environment that I created here. So I've set this up on the Launchpad. This also now makes it accessible to the rest of my team. I'll click Launch. And you'll see here that I'm just going to switch over to the new compute environment that we created. Awesome. And what I'll also do is then define the run output. I'll just say this is pipeline results pilot. And you'll notice here that I have this little toggle in place. Essentially, if I enable this toggle for Run Studios, what this will allow me to do is after that workflow has been completed, I'll be able to actually spin up a Cell by Gene studio with the results of this workflow mounted in. What that allows me to do is basically share that studio with my colleagues, anyone on my team, and hand off the results. Because again, it's not enough to just generate this data and keep it in an S3 bucket. I'll go ahead and launch this. And that should start running. The problem is that when you start launching at scale, that also means more things can go wrong. Tasks can fail. Luckily, the new runs page, the refresh runs page, essentially gives you a really nice view of exactly what went wrong, how analysis was done, and everything at a glance. If I show you the run details page here and the run info, you'll basically be able to see exactly what pipeline from the Launchpad was used for this execution, the history of runs that I might have also kicked off within this workspace, and also be able to preview exactly what compute environment you had used as well. And luckily for computational biologists or wet lab scientists, this view will look the same whether this had run on HPC or different compute environments. So the actual information required to understand how the analysis was done and recreated is all in one place. However, as I mentioned, most of the time things actually go wrong than they go right, and that's fine. Luckily, on the runs page, you'll also get information for a failed task, exactly what container was used, what resources were requested, and what tools and packages were required as well. And what we can actually do is, in terms of kind of like a game changer, I can take all this information and basically spin up a studio's environment that will allow me to recreate this task context. So what I'm going to do is essentially switch over screens. And what you're seeing here is a little Python utility that I've put together. What this utility and script does is essentially make a set of API calls. It's going to call the platform and request information about a failed workflow here that I've provided. And I'm also going to give it the name of a task that failed that I want to kind of debug in Studios. It'll pull information about that task, what container was used, where the work directory lives, what task resources were required, and it'll make another set of API calls to basically spin up a studio in VS Code with the work directory mounted, with the same task resources that were required for that particular task, and the same packages that that task used as well. So I can hop into an environment, debug that task, and continue on with my workflow. So what I'm going to do is I'm going to run this live. All right, so what you just saw, to quickly show you, is that it's going to pull information about the task, where the work directory is, the resources, create a conda environment for me. Specifically, this one failed at the ScanPy task, and then essentially create a Studios environment. Now, if I hop over to Studios, you'll see that I now have this newly kind of built, currently starting environment with the work directory for that task mounted in through Fusion, the same resources that that task had requested when we ran the workflow itself, and all the packages that I'll need to actually debug this task as well. So while this actually starts up, I'm going to hop into an earlier one that I created to kind of show you what this looks like live. All right, and so we're essentially kind of hopping into Studios now. What you'll see on the left side is the task work directory, so all of the .command files required for that task, and also the data that that task required as well. In this workflow, I realized that I unfortunately made a small error in my Python script. Nextflow is looking for an output file with this prefix and a suffix called h5ad. Unfortunately, I just made an error in my Python script and used the wrong suffix. That's okay, we can fix that, and we can essentially do that live. So I realized that I'm writing to the wrong file. So if I fix this, I can then run my command.sh. Oh. And this will then essentially rerun that Python script. And what we should see in a second is a new h5ad file specifically for the UMAP step. So you can see how easy it was to, one, find a task that failed, hop into an environment that recreates that task context, fix and make any changes that I need to my task itself, and then be able to continue on with kind of debugging my workflow. And the key point of this is that you're kind of collapsing the failure to fix cycle from hours to minutes. And you'll see here that it's finished, and I'm able to generate the output that I was actually expecting. Now I can make these changes to my actual workflow and continue on with generating my analyses. So now that we've kind of squashed bugs, we should be able to run our workflow end to end. The problem is that when you're running your analyses, like I said, it's not enough to just keep the file somewhere. Essentially, when the pipeline completes eventually, this now comes down to the real test, sharing results. These H5 files that I'm going to generate are probably too large for email, too complex for non-computational folks to kind of figure out how to visualize them. And so instead what I can do is kind of make it easier for them. Since this workflow is still running, we'll wait for it to kind of kick off. What I'll do is show you an earlier one. At the end of the workflow, what I essentially did was within the onComplete block, I added another set of API calls. Those calls will take the results in the output directory for the workflow, mount them into a custom studio. I've built a custom container to run cell by gene. And essentially what it'll do is mount in those results of the workflow for that pilot data, and then essentially spin up a studio right after. So I'm able to run analyses, explore my results right after, all kind of in one place. Hopefully I can show you this live when the workflow actually starts running, but in the case that it doesn't, you'll see that this one has already created it, and I can essentially go back to Studios and hop in to show you that. And you can see how this makes kind of scaling your teams easier, from fixing a workflow to running a workflow to being able to actually hand off some results to make meaningful biological interpretations from it. So here's an example of that one. It's called a cell by gene studio. And you'll see here I've mounted in the results of a previous run for that pipeline. And I've also specified the memory requirements for it. And here is where I indicated what kind of custom container I wanted to use that will run cell by gene. If I connect into it, what you'll see here is that now we're actually able to visualize the data that we generated. I can click on various clusters to reveal the cell type markers, share this URL with other colleagues and teammates, and actually perform kind of real-time exploration, get questions answered immediately, and not in kind of next week's meeting. So we essentially went from command not found to biological insights and kind of accomplished our mission. I'll quickly kind of go back to the slides now. And the whole point of this is that we went from Friday 3 p.m. crisis mode to Monday 9 a.m. results delivered. Your science should not slow down as your data grows, as your team grows, and your workflows grow. Seqera ecosystem is here to help you, and essentially, we welcome you to doing bioinformatics without bottlenecks. Quick shout-outs. If you would like to use a workflow similar to this, probably don't use our prototype. Instead, use a fully featured one available on the NF core called single cell downstream. And a quick shout out to the team, side of team. These are never individual efforts. And also Seqera for creating solutions that help bioinformaticians tackle these challenges and advance science.